Hey there, everybody, and welcome again to another Insights. My name is Joe Komsky, and I'm the Senior Director of Philanthropy for Sanford Burnham Prebys. Today, I think we have one of the coolest topics we've ever had for Insights, immunotherapy. So, what is immunotherapy? Simply put, it's using our body's own immune system to fight cancer and other diseases. Now, why is this important? The way we treat cancer today is so difficult on the body. If you think about it, radiation, chemotherapy, what we're really trying to do, it's a race. It's a race to kill all the bad stuff before we kill the good stuff. And that is so difficult on a person. And even if we are able to treat and cure the disease, it still is so hard on a body and can lead to complications afterwards. What if we could use our immune system to fight cancer? Our immune systems are such efficient fighting machines, right? They're so great at that. I mean, it may not seem like it, right? When you've got the flu, but think about it. You've got the flu, you've got a virus, you're down for 3 or 4 days, and then we're generally back to normal. Our immune system is really good at that. And it also continues to patrol the body looking for other diseases. There's also a ton of things that our immune systems fix in our body that we don't even know about because it's just going on and doing it. It's repairing cells. It's repairing all the other things in our body that are going wrong. So today we're going to hear from 3 of our foremost experts on immunotherapy about the work that they're doing to help our immune systems fight cancer and other diseases. If they're successful, one day we may be able to eliminate treatments like chemotherapy that are almost as bad as the disease itself. Their work is instrumental in finding more efficient and humane ways to treat illnesses. As always, if you have questions for our speakers, please use the Q&A button on the bottom of the screen. You can do that at any time, and then we will ask questions of our speakers. After each of our scientists speak, we'll have a quick Q&A with them, and then at the end, we'll bring everybody back for questions. So our first speaker today is Dr. Linda Bradley. Dr. Bradley is the Director of Aging, Cancer, and Immuno-Oncology Program at Sanford Burnham Prebys' NCI-designated Cancer Center, and is recognized as a key contributor in the field of CD4 T-cell biology. Dr. Bradley's lab is focused on understanding the regulation of human T-cells in an effort to enhance patient responses to their tumors, and is working to develop biologics for treatments of patients with autoimmunity and cancer. Dr. Bradley, welcome to Insights. Please go ahead.
Thank you, Joe. It's a great pleasure to be here today. Let me get my screen shared and have this opportunity to speak with you. As Joe mentioned, I am an immunologist by training, which means I study the cells of the immune system and how the body defends itself against infections. Joining me today are going to be 2 people from my lab, Dr. Jennifer Hope, who's a postdoctoral fellow, and Dr. Dennis Otero, who is managing our staff scientist, who's managing our pharma programs. So cancer, of course, you all know, is a horrific disease. And here it's showing the estimated new cases in the United States in 2021. And you can see that cancer can affect multiple tissues, and the incidences are different in men and women, but that primarily occurs with the gender-specific diseases such as prostate and breast cancer. But what is cancer? So cancer refers to a collection of more than 200 diseases, and in all cases, this is caused by the abnormal growth and unrestricted growth of cells. So what are cells? Cells are the basic units that make up all the tissues of the body. And in every tissue, the cells are specialized to perform specific functions. And the human body is made of trillions of cells. In all types of cancers, some of the body's cells mutate and begin to divide uncontrollably, and they can then spread to surrounding tissues. Cancer can start in almost anywhere in the body. Immunotherapy is a fairly new approach to treatment of cancer, and immunotherapies are now being tested in almost every and implemented for most types of cancer. But what is immunotherapy? So as Joe alluded to in the beginning, essentially immunotherapy is a term that's applied to any drug, treatment, or therapy that boosts the body's immune response to help it fight off cancer. Often treatments that involve immunotherapy or engaging the immune system are currently being done with traditional cancer treatments such as irradiation and chemotherapy. However, there are many different types of immunotherapy, and responses to immunotherapy can differ with different cancer cell types. And some types of cancers that include, for example, pancreatic cancer and prostate cancer are very poorly responsive to immunotherapy by any approach. So some patients with cancers that include metastatic melanoma, lung cancer, and leukemias respond really well to immunotherapy, but there are always patients that don't respond or patients whose responses relapse and their cancer takes hold again. So a key goal of the research of myself, my lab, and many other people in the field at this time is to optimize and individualize treatments so that one day we may cure cancer in most, if not all, patients. So what is the basis for cancer immunotherapy? Well, this is the immune system, and the immune system is made up of tissues such as lymph nodes, which you probably remember when you get sick and you may have a swollen gland in your neck. That is partly the response of your lymph node, the T cells in your lymph node. The lymph nodes are connected throughout the body by the lymphatic network that's shown here in this cartoon. And the cells that I study in my lab specifically are called T cells, and they are a collection of cells among many different types of white blood cells that specifically patrol the body in search of infectious agents such as viruses and bacteria. When they encounter them in the lymph nodes, they can multiply dramatically and then be released via the lymphatics or via the blood to go back to a tissue where there's a site of infection. And there they work to resolve the infection. And T cells in particular are excellent at killing virally infected cells or cells infected with other intracellular pathogens. Another job of the immune system is to recognize abnormal cells throughout the body that can develop from genetic mutations. So what exactly is immunotherapy? Well, these are examples of immunotherapy that are in the clinics today. So adoptive T cell therapy refers to taking a patient's own T cells from their blood, engineering them to recognize their tumor, and then growing them up to be in the numbering billions and reinfusing those cells into a patient. And this has been very effective particularly for blood cancers, but not for other cancers to date. Another type of immunotherapy we call nonspecific. And here I've listed two proteins that are products of immune cells of the immune system, that have immune protein and T cell or immunomodulating, enhancing effects. There we also have being tried what's called oncolytic virus therapy. In this case, has only been treated so far with melanoma, where a common cold virus called herpes is used to inject a tumor specifically, directly. The virus replicates only in those cells, not in normal cells, thereby killing the cancer. And then there's great hope for cancer vaccines. I think most people know about the preventative vaccine for human papillomavirus, which can ultimately lead to at least 6 different cancers later in life. And that's really the star so far for preventative vaccines. Treatment vaccines are fewer, but are being widely studied. And in this case, the goal is to utilize agents that will help jumpstart the immune system while cancer is already on board. But many of you have probably heard about immune checkpoint blockade or monoclonal antibody treatment. Some of these treatments are even advertised on television. So in 2018, the Nobel Prize for Medicine was given to two individuals in the field of immunology who discovered PD-1 and CTLA-4 over the past 2 decades. Now, these treatments are given to patients routinely for certain cancers now, such as malignant melanoma and non-small cell lung cancer. And they are also being tried in virtually every type of cancer because of their very profound efficacy in a few patients. So unfortunately for the field, the monoclonal antibody treatments or immune checkpoint blockade is not it's not the end of the line and will work for everyone. So each individual cancer can be detected at a different stage, and some cancers develop mechanisms to prevent the immune response from getting underway. So our laboratory is studying T cells, as we mentioned earlier on. We initially started in my work many years ago studying immune responses to viruses. And it was the hope back then that we could harness the immune system to cure cancer. And in fact, most of you are probably not old enough to remember Nixon's war on cancer, and the immunologists were going to figure out how to cure it. Well, today we are making great strides in this. This is showing you now T cells in blue and a cancer cell in red, and the T cells are ganging up on the tumor cells and they're directly killing them. And I'm going to show you a video which I think shows the amazing capacity of the T cells to kill tumor cells. So the T cells are the smaller, rounder cells that are climbing all over the elongated tumor cell. And when you see a cell bubbling up like that, that's a process called apoptosis. And a T cell can kill over and over again. So one of the things that my lab is doing is figuring out alternative ways to harness the T cell, the function of T cells to kill tumor cells and either add to or improve the existing immunotherapies or become novel therapy in and of its own.
Okay, we had some comments before that some of you couldn't see me when I was on screen. Trust me, if that's the case, you're not missing anything. Linda, thank you. I have a couple questions. And again, if people have questions, please put them in the Q&A box at the bottom. But let's start with the basics, right? So you just showed a video, which was great, with T cells killing immune cells. It looks so fantastic. Why don't our bodies do that already? How are the cancer cells avoiding our T cells now?
Well, that actually is the topic of discussion that we'll just follow by Jennifer Hope. She is going to tell you exactly what happens to T cells in the context of cancer, which makes them unable to respond, and how the immune checkpoint blockade specifically can reinvigorate the immune system. So in essence, as Jennifer will explain to you, the T cells try to kill the tumor cells. The tumor cells can mutate and not be recognized by the immune system again. And the other thing happens is they get worn out, in essence. They get tired and they can't function. So those are the two ways that the immune system is usurped by the cancer, either immune escape, it's called, or T cell exhaustion.
Thank you. And when I introduce Jenna, I will apologize for trying to usurp her her talk with my question. We do have a question from the audience about nutrition. And does a patient's nutrition make a difference in how their body responds to immunotherapy?
Well, immunotherapy is actually, one would need to know, is not a simple treatment that doesn't have side effects. It has profound side effects because you're unleashing the immune system when you take the brakes off. So an individual can feel quite ill, potentially even as ill as on chemotherapy, because you're really disrupting the body's homeostasis. So anything a person can do to maintain their health during the process of treatment, which would include good nutrition, getting enough rest, all of those things are absolutely critically important. And there is a whole school of thought that if you have cancer and if you're undergoing treatment, it is absolutely critical to maintain good nutrition.
So the idea of unleashing the immune system, it seems to me that that might put someone in danger of developing, let's say, an autoimmune disease. So it's important, I would imagine, to find that balance between
Right.
energizing our immune systems, but not letting it run rampant and killing the healthy parts of our body.
Right. So it's unfortunate that autoimmunity, which is the body's immune system to attacking its own cells and tissues, is a side effect of immunotherapy, particularly the immune checkpoint blockades, which are not specific to a particular tissue. And what happens then is actually the patients are monitored carefully for signs of autoimmunity and inflammation. And those are at the moment controllable, or these are called adverse events, and they are definitely part of immunotherapy. And most clinicians feel that treatments with things like steroids or other things that dampen inflammation are able to make the treatments manageable for the patients so that you can continue on the treatment. It is true that the regimen's different for the different monoclonal antibodies, and the anti-CTLA-4 is very toxic, so it's only given once or twice, whereas the anti-PD-1 treatment is less so, and people receive multiple doses at like every 2-3 week intervals because it's much better tolerated. So these things are actually very empirical in terms of what the doctor and the clinician are evaluating, and they make changes. It can make changes and do make changes depending on how well a patient is tolerating the therapy. But autoimmunity so far has been, with the currently available treatments, has been manageable for most patients by standard anti-inflammatory drugs.
Great. Thank you, Linda. We're going to move on now to keep things moving, but we'll bring you back at the end for other questions. And so now I'd like to introduce Dr. Jenna Hope. Jenna, again, I'm sorry for jumping the gun with your presentation with my question to Linda. Jenna is an American Cancer Society postdoctoral fellow in Dr. Bradley's lab at Sanford Burnham Prebys, also within the Aging Cancer and Immuno-Oncology program. And we'd like to thank the American Cancer Society for supporting her fellowship. Her research interests are centered on discovering new ways by which we can use our immune system to fight off cancer and infection. Dr. Hope, the Zoom room is yours.
Thank you, Joe. And it's not a problem at all. It's an excellent question in terms of, why can't our T cells necessarily function the way that we would want them to? So I'm super excited to share that with you all. It's really a pleasure to be here today with everyone, and to really just, especially Linda and Dennis, and the opportunity to share with you all what it is that we are interested in when we're talking about cancer, and particularly cancer immunotherapy. So I'm just going to bring up my mouse here. So when we talk about the immune response to cancer, as Linda was speaking about, really in Linda's lab, what we're focusing on are T cell responses. And in particular, we're talking about the abilities of these T cells to really go after and directly kill tumor cells. And actually, Joe, you brought up a great analogy in the beginning, and you didn't even know I was going to talk about it, but I like to think about this in comparison to influenza all the time. So as you can imagine, this whole process of us and our cells, our immune system fighting against cancer requires energy. And the analogy I like to think of is if we're going to the gym for a workout or going outside for a run. And so our immune system is pretty well developed in the respect that if we see an infection, such as a viral infection like influenza, our T cells can fight that off. It's basically akin to us going out there with or without some training and being able to run a mile or maybe even a 5K. We're going to be tired at the end of it, but we can handle it. And when our T cells or our immune system is facing a viral infection like influenza, as you suggested, they can get to that finish line. They can kill all of the virally infected cells. But when we're talking about cancer, this is more like going out there and trying to run a marathon without any of the specialized training that we know marathon runners need to take. This costs our cells so much energy that many of them become exhausted, as Linda talked about. That's an actual term that we use to describe these cells. They become exhausted, and many of them cannot make it to that finish line. And so that's really the problem that we're facing. But it's even more than that, right? So our T cells, they're racing an uphill battle. So it's not just a marathon anymore, right? But now it's a marathon that you're having to run up this hill. And in particular, it's not just even that. So our T cells, oftentimes, once they get into the actual site of a tumor, in the beginning, it's like running that 5K. They're full of energy, they're there to attack and kill the tumor cells, and they do a really good job about it. But the tumor cells, like Linda alluded to, are also trying to outcompete the T cells in our immune system. And they have this special ability to really change the whole environment to make it such that it's not good for our T cells or our immune cells. And so what we've been working on in the Bradley lab is this immunotherapy target. It's a novel one that we've been really excited about for the last several years. And what we have found is that our T cells express this target pretty much all the time. But when they get activated, meaning when they see a virally infected cell or a tumor cell, they start to express even more of this target. It just kind of hangs out on the surface of this cell. And so we've really been studying what it does. And what we found is that this target has the ability to basically bump into other things going on in the tumor microenvironment. And as it does that, it's basically adding extra weight to our T cells. So now, not just is it a marathon, not just is it an uphill battle, but we're adding on weight to that backpack, and our T cells are having to carry it with them. So you can understand how that could take a lot of energy and really exhaust our T cells even faster. But the really great news and the exciting research that we found is that we can actually block this target. We have models where we've removed this target from the surface of T cells. And by doing so, we're really limiting or preventing the development of that exhaustion of the T cells. Not only that, but we're giving them this extra boost of energy. They're able to get up the hill faster. And what's really important is that we have found in our lab that targeting this particular target on T cells helps us to limit growth of tumors that are currently resistant to the checkpoint blockades that are available. So that's really important because we don't fully understand why some patients respond really well to current therapies, but why many of these patients don't respond. And so we want to make sure that there's a lot of different options out there for patients so that if they can't respond to a current therapy, maybe this new therapy is instead a different answer for them. Or in some cases, maybe we can combine it. And by that combination, help to target those tumors that are really difficult to treat currently. So that's been the focus. We find that by targeting this particular molecule on the surface of our T cells, we're really unleashing that immune system. We're taking away the brake and giving our cells that extra burst so that they can fight that fight and cross the finish line.
Thank you, Jenna. We have a bunch of questions here, but let me start with this because cancer and viruses, they can evolve and they can change. And so how are our T cells, are they able to adapt to that evolution, or does that help the cancer sort of hide from our immune systems and continue to hurt us?
Well, the cool thing about T cells, without going into too much detail, is that T cells really recognize the guts of something, if you will. So our immune system is really diverse. It's made up of a lot of different cell types, and each different cell type has a very important role to play in targeting different things. So when we think about a virus infection, like you talked about mutations of viruses, oftentimes we're first thinking about surface changes, right? That's going to be handled by a totally different type of immune cell, a B cell, and antibody responses. Our T cells are looking at the inside of the virus. That is less likely to change when we're talking about a virus. When it comes to tumors, the biggest difficulty our immune system faces is the fact, like was alluded to earlier, there are normal cells that something has mutated. And it's, again, that balance between a strong immune response and prevention of autoimmunity that we want to maintain, that fine balance that we don't attack normal healthy cells. So if a tumor mutates in such a way that it's not recognized as a damaged cell, then it can evade a T cell response. And so it's, I'm not sure if that fully addresses your question, but that's one of the difficulties that our T cell faces, is making sure that it can recognize it, but also recognize that it's different enough that it needs to be killed.
So does that mean we may need to do, I know, for example, CAR T therapy is sort of engineering T cells to recognize proteins, et cetera, markers on tumors. Do we need to do some combination perhaps of that plus the checkpoint inhibitors to be able to do, for example, like a vaccine?
That would be a fantastic approach. The limitation is we have to know what the target is. And in some cases, we don't know what it is that the T cell is recognizing. So there's a lot of work going into, in general, in the field with neoantigens. That's a term that we give for these new molecules that pop up on tumor cells that our T cells can recognize as being what we call non-self. So it's different than what our normal body would express. But in order for a CAR T cell to work, we have to know what the target is. And not just do we have to know what the target is, but that's one of the dangers of the CAR T cells too. We have to make sure that it's specific. That's why, for example, with leukemias,
Okay, sorry, I think we lost Jenna for a second. Okay, so Jenna, you froze. So we have some good questions for you though, so hopefully we will get you back. And in the meantime, let's go with Dr. Dennis Otero. Dennis is a staff scientist and project manager in Dr. Bradley's lab, and his interest revolves around the discovery of biologics, which can regulate the immune system to fight infectious disease and cancer or to prevent autoimmunity. Dr. Otero, thank you for joining us today. Please go ahead.
Thank you. I'm going to go ahead and share my screen now. Thank you for the introduction. What I am here for is to tell you what happens next, or where do we go from here. Now in Linda's lab, I actually wear two hats. One, I am a staff scientist, a research scientist that conducts research and performs experiments. And I am involved in many of the projects within the lab, lending my expertise to much of those projects. But what I also am is a project manager. And what I do is manage multiple projects in translational science. So what is translational science? So much of what we do at universities and research institutes is called basic science. And that is, we're trying to ask the how questions and the why questions and come up with answers to those. But every once in a while, we'll have discoveries like our target that Jenna mentioned earlier that have a direct relation to human disease, in which case we want to translate our discoveries into maybe new therapies for that human disease. So what usually happens is after we publish our discovery, pharmaceutical companies will read those manuscripts and take those ideas and try to make drugs out of them. What we're trying to do here at SBP is actually take those discoveries that we made ourselves and carry them to the clinic, kind of a bench to the bedside type of structure. So like Jenna said, we have a target now, and we've done our basic research. Most of our basic research happens in mice models because mice have a very similar immune response as humans. But now we need to translate that over to human immunology, in which case we have to start studying patient samples. So as project manager, and what I need to do is collaborate with clinicians, doctors in the clinic who work on human patients and obtain the tumors cells and patients' T cells from the blood from those patients. And then we can then try to associate our target, see if our target in the human cells behaves the same way as mouse cells. Now, we are currently undergoing that, as well as developing therapeutics for our target. So since this is a protein that is expressed on the surface of a T cell, it was really, we'll be able to generate antibodies towards that protein and try to block our target. So right now we have collaborations with a biotech company to produce antibodies. They have dozens of antibodies currently towards our target that we will start testing for the ability to bind the target cells and to block that target molecule. From that point on, everything becomes a little more complicated. We have to start clinical trials, which evaluate safety and the toxicity of our compounds that we develop. There are companies that we will collaborate with in order to do those as well. We will then start to evaluate efficacy of our product as well. And that will be going back to the clinic, going back to the doctors in the clinic, and collaborating with them to start clinical trials to evaluate efficacy. And once we have a molecule that we think is working as well as it can, then we can go to our next phase and start comparing that molecule to what's already being done in the clinic to treat those cancers. And that's how we go from starting out with basic science and then translating it to human disease.
Dennis, that's really interesting, and thank you for sharing that with us. How often do things sort of, they work in the lab, they work in a mouse, but then it doesn't always translate to a human? Things sort of fall out of those trials. Is that a high percentage, a low percentage?
I'd say it's probably a high percentage. We cannot be sure because, like I said, we're not a drug company, so we don't know what the failure rate of those is. But yes, definitely not everything translates going from mouse to human. There, of course, are major differences, but the mice are similar enough that we can do studies and gain a lot of insight from them.
We have a question from our audience. Is the phase 1 trial only with human blood and not people?
The phase 1 trials, I believe, are, they start off with the safety, the toxicity studies are with animals. So I think they will start with closely related species, monkeys, and then maybe apes.
Got it. Okay, let's bring everybody back because we have a bunch of questions. We are very happy that we have Jenna back. Jenna leaving us, I just want to say, and congratulations are almost in order. Jenna is, I'm going to say, days away from giving birth to your first child. So I'm glad that it was just computer freezing and not having to rush to the hospital in labor.
No, sorry about that.
No. That's quite all right. That's quite all right. So let me throw this question out, and for probably either Linda or Jenna, how is the correct immunotherapy identified for patients?
Linda, do you want to take this
I
one?
can take part of it. Maybe you can jump in if I miss something. Well, actually, there's not a good way. Jen and I work with a melanoma skin cancer oncologist. And one day I was talking about the project with him, and a young oncology fellow comes into the office and asks about starting a new drug. And it's a monoclonal antibody to a target that I'm aware of. And I said, oh, how did you make a decision to start the patient on that treatment? And he said, well, we have a trial, we don't know. And so there's a huge effort for every one of these checkpoint blockade agents and there are hundreds of trials. If anyone wants to check this out, go on trials.gov. And everyone is looking for biomarkers. That's what they call a marker that suggests or would be correlated with a good response. So for PD-1, which, the presence of PD-1 is necessary for anti-PD-1 to work. Also, the ligand or the molecule that PD-1 interacts, PD-L1 for PD-1 ligand, people have looked, well, is that going to be something that marks a tumor that's going to be responsive? Unfortunately, the answer is no. And that there is nowadays a really, I would call system-wide approaches, bioinformatic approaches that take tumors and interrogate hundreds of proteins and genes within cells of the tumor to see if there are any collection of proteins or genes that could be correlated or indicative of a patient's response to an immunotherapy. At present, there really isn't a really good biomarker for any of these treatments. And unfortunately, for patients whose tumors are very responsive, such as melanoma patients, there's an estimated 80% of patients that initially respond, but less than 20% have what they call a durable response, meaning a response that goes out at least 5 years. So the immune system also becomes refractory to treatments, which is yet another problem. So at the moment, I think that the approaches that are being widely accepted as potentially valuable for improving patient outcomes are to combine various therapies and try to then prevent this, the loss of responsiveness. So come in with a new agent at a later time that blocks a different pathway. And one of the interesting things that our field is very, very focused on right now is there are multiple inhibitory pathways in the immune system. And the reason is it's so important to tightly control immune responses that all of those are acting as checks and balances, and often at different times during an immune response. So the big problem is we're looking for biomarkers, and there are publications that are coming out very often now with these large bioinformatic approaches to do multiple gene analyses, hundreds of thousands of gene analyses, hundreds of thousands of protein analyses to try to come up with networks or groups of proteins or groups of gene that might help us one day know exactly what, and every patient is different, unfortunately. So you've probably heard of personalized medicine, and at this point, that is where things will go. We will actually take a slice of the patient's tumor and do these advanced technologies to look at not just one or two biomarkers, but a constellation of things. And I think we will get there, but it's a big challenge.
The only thing I'd like to add to that is just the fact that, again, remember, we're not clinicians, we're scientists, we're looking at from the literature side, but, keeping in mind that the physicians who are making these decisions, a partial response could mean the difference of a couple more months or a year for that patient. And that could be a huge change in their quality of life or what they want to do. And that's why it's such an important conversation for that patient to have with their clinician who knows their individual case history and everything else. But as Linda says, there's not a one size fits all. It's important for everyone, I think, to understand how Linda mentioned, like, cancer is multifaceted. It's not just is it a collection of 200 different diseases, but even if you have melanoma, there's different subtypes. And every patient basically becomes a unique case. So that's why it's just so important to have those important conversations with your physician and make the best judgment call for yourself.
Thank you. We've gotten a couple questions from people who have said that they themselves may have autoimmune diseases and wondering how safe immunotherapy would be for someone who already has or has had autoimmune diseases.
I'll take this because I have an autoimmune disease. And if I had cancer, I wouldn't personally have any concerns about taking an immunotherapy. Understanding, my particular disease and also, what these immunotherapies do. The monoclonal antibody treatments specifically, if you remove treatment, it removes the effect, right? So it's a transient treatment. And again, it's going to depend on a person's specific case, but an autoimmune disease, for the most part, is really an overreaction of the immune system or a reaction that shouldn't be taking place. That could potentially help you, right? If you have some extra inflammatory response going on, that might rev up something else. It's going to depend on the specific disease, but at least personally speaking, that wouldn't bother me at all.
Got it. There was a question here. I'm going to read it specifically. Would the immunotherapy research you are performing be generally similar to the treatment performed today for plaque psoriasis and targeting IL-17A? Linda, maybe.
So IL-17A is what's called an inflammatory cytokine. So it's a protein that's produced by immune cells, including T cells and specific subsets of T cells. So the treatment that blocks IL-17A is blocking a secreted product, a cellular secreted product. The target that we're looking on is actually expressed on the cell surface. So in that way, they would be very different. Now, one could also take an alternative approach, and I don't know if this is the clinic. Again, we're not clinicians, but there is a receptor, of course, a protein that binds IL-17A, and you could block the IL-17 receptor, and that would be another approach. So it's like there are a number of approaches, and that would be expressed on the cell surface. I'm not at all sure whether there are any anti-IL-17 drugs on the market, but the IL-17 drug, 17A drug, I think has been very successful for psoriasis, to my knowledge. Dennis may know better. He's worked more on that side of things.
Yeah, so I would just add that these are opposites, right? In our case with the cancer, we're trying to boost the immune response in order to clear the cancer. On the autoimmune side, we're trying to dampen the immune response. That's what those anti-IL-17 are, dampening the inflammation to block the plaque psoriasis.
A couple questions in general about things that might affect the immune system and or cancer. And again, I know we're not clinicians, but I will ask, I'll bring a couple of these up and perhaps we can tackle them if we feel like we are expert enough. And if we feel like we're not, then the answer will be discussed with your physician. How does the intake of alcohol affect cancer?
Well, I don't know that. I know that it can cause cancer, as far as I know, but it's not something I'm familiar with.
Okay. And as we have said on other things, for anybody asking these questions, everything in moderation, right?
Right. There you go.
Okay. the effect of fasting, because we've talked about that and promoting autophagy and things like that. What effect does that have on our immune system's function?
So that's actually really interesting. There's a couple different, fields going on, and there's a big difference between chronic fasting and chronic caloric restriction to the point of, malnutrition. And that has a really negative effect on the immune system, as you can imagine. Whereas there's been some studies that have shown that intermittent fasting can help promote immune system regeneration. But to my knowledge, those studies have been looking at the immune responses as a whole, not necessarily under the context of cancer. I would imagine, again, we're talking about, in my section, I was talking about how much energy our cells require to continually fight off virally infected cells or cancer cells. We talk about when you have a cold, make sure you stay hydrated, make sure you try to eat, broth or something to give yourself some nutrients there. That I would, not having seen any of these studies, but I would imagine that, potentially caloric restriction could be a downside in that case. I haven't seen those studies though.
And I would imagine that the opposite of that may be true as well. And that's another question we have here about stress and weight. And I would imagine that we were talking about excess weight having on our T cells' ability to fight disease and protect our body. Would that be true?
I would imagine so. I mean, we don't work with obese mice. Those model systems do exist. I'm not sure if, Dennis or Linda, you're, aware of any studies looking at that.
Well, I serve on NIH study panel, which reviews grant applications, and some of these types of study, and it's for basic research. So I'm not evaluating proposals for clinical trials. But Jenna mentioned earlier on the flu, and there was a group that was very interested in how obesity would impact the immune response to flu, and it had a very profound impact. And this particular group was interested in determining, since obesity seems to be a risk factor for the coronavirus disease, that whether or not this would impact the immune system as well. It's also important to realize that I think as immunologists, we view obesity as a chronic inflammatory disease. So there actually are T cells that can be found within fat tissues. And it is without question that obesity does impact immune responses. But I'm primarily aware of particularly this one set of studies because I was interested in I was part of the grant review. And I would say, yes, being a healthy weight is going to be important at any point when you get an infection or cancer. And it is a risk factor that can impair the immune system.
And then a question specifically, what type of immunotherapy is IVIG?
So that would be, I'm assuming, intravenous immunoglobulin.
Yes.
That's going to be basically a replacement therapy, typically somebody who has a B cell deficiency, usually a primary immunodeficiency. So something genetically has happened in them that makes them unable to produce those types of cells that make those antibodies that help protect us against infection. And then actually that's, I think, where I got cut off before, but that's something that you can do in replacement therapy for CAR T cells with leukemias, is that not only does that kill all the cancer cells, but it kills all your healthy B cells too. And so IVIG would be a replacement therapy then to basically infuse these antibodies into your system, and it gives you, an intermediate protection for, I believe, a month is about standard for IVIG.
Got it. Well, thank you all. Thank you to all of our presenters, and thank you all for joining us. As always, our presentation was recorded and will be emailed to everyone who registered. And we are very, very appreciative that you take an active role in learning about our research. Please feel free to share it with others who might be interested in this work. You can learn about our research at future Insights events, and we're going to put up a slide, here it is, of a list of those upcoming events in the next few months. And if you'd like to learn more about or contribute to any of our research, please contact me at either the email or phone number on your screen. I'd be happy to chat with you about how you can help our scientists do this amazing work. Thank you again for attending. And again, please feel free to share with all of your friends and colleagues. And thank you for your interest in our science at Sanford Burnham Prebys. Have a great day.